Hello, I'm Rodrigo Pantoni, and I'm going to present a project, Insights of Forensic Identification of Shrimps Commercialized in the State of São Paulo, developed by me, João Pantaleão, and Fernando Mantelatto. We are from the Laboratory of Bioecology and Crustacean Systematics, the LBSC, from the University of São Paulo in Ribeirão Preto. The topics of this presentation are introduction and objectives, development, results and discussion, and conclusion. In Brazil, shrimp are marketed in label packages that must comply with Normative Instruction number 23 of August 20th, 2019. With this, our objective is to test the accuracy of the information on the identification of shrimp on the label in the package through mitochondrial molecular markers, such as the 16S gene. For this, some packages with shrimps of different species, such as white shrimp, pink shrimp, and sea bob shrimp, were acquired as samples, and about 10 specimens of each were chosen for molecular analysis. Then, the DNA of these specimens was extracted, amplified by means of the polymerase chain reaction, purified, and sent for sequencing. The sequences obtained were entered into the Geneious Prime software for editing, in comparison with sequences already deposited in the GenBank database via the BLAST tool. We found that there is imprecise labeling, that is, error in species identification. In one sample, we found specimen of the species Pleoticus milleri of the Solenoceridae family, which should only contain pink shrimp, species of the genus Farfantipneus of the Penaeidae family. In this case, at least 40% of the specimens in the sample were misidentified. However, the other 14 samples were in accordance with the regulations regarding species identification, in terms of the specimen selected for analysis. One of them presented the information mix of species, and the 3 specimens sequenced were sea bob shrimp. This figure illustrates graph A, which concerns the efficiency of identification of specimens of each shrimp species treated by the popular name. Graph B shows the efficiency of identification among the samples of each shrimp species. We can see that no error was found in the identification of the species Lithopneus vanamei, the white shrimp, nor of Schiphopneus creyeri, the sea bob shrimp. The numbers above the bars correspond to the number of specimens and samples of each classification correctly identified by the label. The Pleoticus milleri can be justified by one of 2 hypotheses proposed here, or by the sum of them. Considering that the manufacturer of the sample that presented this error is from Santa Catarina, a state covered by the distribution of the species, that this species is abundant in the region, and that its exploitation has increased, it can be assumed that some P. milleri were captured there, processed, and marketed as pink shrimp. Alternatively, during packaging processing, representatives of P. milleri may have been confused with pink shrimp due to the reddish color characteristic of this shrimp. The lack of knowledge of the scientific names of the species in question may contribute to this confusion. The absence of errors in the identification of Lithopneus vanamei can be explained by the fact that most samples come from the cultivation of the species, in which there is a greater control of the individuals cultivated and processed for commercialization. Thus, the identified provenance seems to be a condition that reduces the risk of error and/or mixing of commercialized species. The lack of precision in the identification of pink shrimp found in our study gives rise to the proposition of recommendations relying with consumer rights and species conservation, in particular with regard to the correct identification of commercialized shrimp, such as the hiring of biologists by companies as cell shrimp to perform this function. These are the references used in the development of the study. If you want to look at any in particular, it would be nice to pause the video. I would like to thank you for your attention and thank the members of the LBSC. Furthermore, I would like to thank the funders who made the project possible, UB, FAPESP, and CNPq.